for our last biochemical test, we are going to test for tryptophan hydrolysis, so the Indole test. So we're going to make sure that we sterilize our benchtop and sterilize our inoculating loop, and we're going to put it into the microincinerator for 10 seconds. Once the 10 seconds is over, we're going to pull it out and let it cool for about a minute, and we are going to flick the culture broth test tube, make sure it's thoroughly mixed so that we can get a good sample from the test tube. We're going to open the test tube and we're going to hold the test tube lip or test tube opening, hold it to the microincinerator here for a couple 10-20 seconds to make sure that the test tube lip is sterilized. Once it is, we are going to put our inoculating loop in and we're going to swirl around to get a sample onto our inoculating loop. And before we do anything else, we want to make sure that we sterilize the test tube lip again to the microincinerator by holding it to the opening here. And then once that test tube lip is sterilized, we're going to close the test tube. And then we're going to repeat the same process here. We're going to open the test tube, take the cap off. We're going to hold the test tube lip to the microincinerator to sterilize it before we put our inoculating loop with our samples into this broth. Once that happens, we are going to dip our inoculating loop into the solution of the broth, and we're going to do this motion here just so that our sample of bacteria can really interact with the tryptophan broth. Okay, so once that happens, we are going to sterilize the inoculating, not the inoculating loop. We're going to sterilize the test tube lip of this test tube, put it to the edge here of the microincinerator, and then close the cap. We are going to gently swirl the test tube just to make sure everything is nicely mixed with the culture that we just put in. Next, we're going to grab Kovacs reagent here, and we're going to add it to this tryptophan broth with our culture in here. We're only going to be adding 10 drops, which would be enough to react with the sample that we have here. And we're going to mix gently by rotating the tube between our palms. And so after we do that, we will either get a positive or negative test. If we get a positive, we will see a cherry red ring at the surface here. And that will tell us that there is indole in there for the Kovacs reagent to react with. If it is negative, we will see a green or yellow ring at the surface instead.